joining us. Hello everybody, thank you for joining us for the seminar today. I'm Charlie Clifford and I'm going to be discussing rodent health surveillance innovation driving changes. I work for a company, Charles River. We sell laboratory rodents, and we also run a commercial diagnostic lab. So we do the monitoring not only for our own animals, but quite a bit of outside monitoring for externally submitted animals as well. And I work to develop our own internal health monitoring programs to be sure that we know what's in our colonies, we're not missing anything, that sort of thing. We can't afford to have false positives or false negatives. And then I also work with a lot of our external clients trying to help them improve or optimize their own health surveillance programs. I'm sure everybody is very familiar with the problems that are incumbent on health surveillance programs today or inherent to them. As we make more and more precise measurements in science, we find that there's an increasing need for microbiologic stability. Let me just stop for a moment here and say, if you have questions as we go through, please type them in, and if possible, I'll answer some as we go, but I'll get to any others at the end. So please remember to enter your questions. So there is a need for increasing microbiologic stability of the animals that we use. Some people are looking at the microbiome, and certainly those animals need to be microbiologically stable. Also, we're using a lot of animals with immune deficiencies, both to study the immune system and to grow human tumors or other tissue components. These have increasingly more and more severe immune deficiencies, such as the NSG mouse. In the face of this need for microbiologic stability, though, there are some subclinical infections that remain quite common, and I'll touch on those. And one of the reasons I think they stay so common is that it's the negative side of using individually ventilated caging. Individually ventilated cages are used to prevent infections from going from one cage to another, and they work very well, but they're not perfect. And so it's still possible that one or a few cages on a rack could have some sort of infection or another. And that makes health monitoring, or HM, very difficult. Sometimes even infections could get into one or a couple cages and then just die out. Maybe your sentinel gets positive, and by then the infection has died out on the rest of the rack. And the way that everybody uses to monitor these individually ventilated cages is a soil bedding system. And we'll talk a little more about that. And although it works okay much of the time, it does have some significant limitations that I think everybody needs to recognize. And then finally, everybody is working in an environment of increased economic constraints. Everybody wants to save money. Everybody wants to get more for the money they're spending. And that's just the reality. And that has a downside, but it also has the good side of continually pushing us to try to improve. So there are some solutions to some of these, obviously, and that's what I'm going to be talking about today. There have been some improvements in serology. Two of them, I think, are that miniaturization of the assays has permitted increased testing that can be done with very small amounts of blood. And that allows us to take a tiny bit of blood from an animal without having to kill the animal just to see what it had when it was alive. One of the ways that we do this now is what's called dried blood spot sampling, where a tiny spot of blood is just put onto a filter card, and then that can just be sent in the mail for testing. Also, PCR is used more and more, and it has been enhanced to where now we can just test animals directly coming into a quarantine facility to see what active infections they might have that could pose a threat to the other animals in the facility. And by testing these directly without having to use sentinels, it can accelerate the whole process. You can get perhaps 3 or 4 times as many animals through quarantine as you could previously. And finally, sensitivity of PCR has been developed to the point that we can just sample dust in the animal's space without even having to sample the animals at all. So we can just look at the dust and we can tell what infections are present in the animals. And finally, I think people are increasingly recognizing that quality control or health monitoring is just a form of quality control to see if the animals meet your specifications. It's not some sort of vague, take a look at them, see what else they might have. It's to see if they have a very specific list of agents. And by doing it that way, we can design tests for each of those agents, and then you can just ship samples. You don't even have to ship animals at all. So we'll go through each of these. So I mentioned that there are some infections that are still common in mice, and we may have an international participation today, and these are some numbers from around. In Europe, the numbers I've seen are very similar, but essentially laboratory mice tend to have the same infections everywhere. They have norovirus, the various parvoviruses, mouse hepatitis virus, epizootic diarrhea of infant mice, EDIM, and TMEV, or Tyler's murine encephalomyelitis virus. I did some compilation of numbers of just coming through our lab. So these are all the samples that have come through our lab in the last several years. It's about 800,000 samples altogether. And I put prevalence in quotes because it's not really prevalence as in this is the percent of animals in the vivarium that have the infections, but you know, the things that we see at a very high number, those are common. And the things that we don't see, if we haven't seen them in 800,000 samples, they're really, really rare. So what should you worry about? What's most likely to get into your facility? What's the stuff that's common? The same things that are getting into other people's facilities. And so you have to take these numbers with a grain of salt. We see a lot of norovirus. We see quite a bit of the parvoviruses, MPV and MVM. But even though only about 1% of the samples that come through our lab are positive, if you get out and you ask people at different institutions whether they have it at their facility, it turns out that probably 40% or more are positive for that. So it's a very high number of the institutions have at least a little bit of parvoviruses. And then the viruses we monitored by serology, MFIA, and Helicobacter, Pasteurella pneumotropica, fur mites, and pinworms, those are numbers that we produced by testing by PCR. So it's a little bit different. Just have to keep in mind, those are the numbers we're finding. But those are the things that are common. So how do people check for these? Well, in North America at least, in most research facilities, probably the most common way to house mice are in these individually ventilated cages. And since something can be present in infection, could be present in those, it may be only 1% prevalence, you really can't sample enough animals randomly to have a good idea if there's anything there or not. So what everybody does is they use soiled bedding sentinels. And the usual scenario for this is there will be one cage of 2 mice per side of the rack. And one side of the rack might have 80 cages. So 1 cage of sentinels for up to 80 cages of mice, the research mice. Every week, when the bedding in the cages gets changed, 80, even 100 cages sometimes, about 5 mils of bedding once a week gets moved into the sentinel cage. That's about a teaspoon of bedding. The cage is generally changed in a hood. The soiled bedding gets put in either into a paper bag, as I've heard some places, or just into what will be the sentinel cage. And then at the end of the day, the sentinels get that bedding. If there's too much bedding, they may only get some of it, and hopefully it gets mixed up. So there's a lot of dilution there if only one of the cages had any infectious material in it. So what do we assume? You know, we're assuming some things are happening during the monitoring period. And the monitoring period is usually 3 months. We're assuming that whatever they have is going to be present in the feces and gets transferred into the soiled bedding. We're assuming that the people doing the transfer are actually collecting some fecal pellets and not the cleanest looking bedding. You know, and maybe we get some things that could be shed in the urine, but not much is shed in the urine. We assume that the sentinel actually gets an infectious dose into its cage. And remember, for some things, it takes a pretty good dose. Parvoviruses, for example, a few viral particles are not likely to cause an infection. So we're assuming, though, that the sentinel cage will actually get an infectious dose of the virus. We're assuming that whatever it is we're trying to transfer doesn't just dry out and die before it actually gets to be exposed to the sentinel. And we know for Helicobacter, it can die maybe in half an hour or so. So we're kind of working against ourselves because we're assuming that whatever we're transferring wouldn't dry out. We're assuming that even if there are only a couple fecal pellets that are infectious in that entire sentinel cage, the sentinel is going to find that particular fecal pellet and infect itself. And I think it works pretty well. It's not bad, but we're assuming that happens. And really it does work much of the time. With all those assumptions, you can see why it doesn't work all the time, but it works pretty well much of the time. But it does have some limitations. So if we take that previous chart, or graph, or table rather, and we look at what actually gets transferred very reliably by soiled bedding versus what only gets transferred sometimes or really very poorly, we can see that most of the viruses get transferred pretty well. We know norovirus doesn't always get transferred, but obviously it does pretty well, or we wouldn't be seeing 40% of the animals, the samples that come through our lab be positive for it. But we know sometimes it's not. Helicobacter tends to dry out. Termites may not be present in the soil bedding. Pinworms, some of the pinworms are not even present in fecal pellets, so those don't get transferred very well. So the conclusions for the sentinels are that it can be successful for some common infections, and sometimes it finds others, you know. Sometimes you can find Helicobacter. Sometimes you can find pinworms or termites even, but not so often. Still, if you're using soil bedding sentinels over a long period of time, sooner or later you're likely to find what's there. And if you're using it across your entire facility, you probably have a pretty good idea of what shows up now and again in your facility. But it's harder to know what's on a single rack at a given time because sentinels are just a little more fuzzy. They're not as focused. But until recently, this has been the best method available. So let's look at what we have for diagnostics pretty briefly. Serology is the old mainstay, and it's a beautiful thing. It really is. Serology is great. It's inexpensive. It's highly specific. It seems to be very sensitive. We don't get a lot of false positives. We don't get too many false negatives if the animals have a good immune system. It takes about a week for an animal to produce antibodies, but after that, they're present for the life of the animal. And so that's really good. It doesn't matter whether a virus was in the lung or the liver or the kidney or whatever, we know what sample to collect. We don't have to go looking for the agent because it will always be present in the serum. And so that also is very good. One of the disadvantages is that we can only test one animal at a time. So, but for the animal that we're testing, it really works very well. As we developed larger and larger serology panels, trying to test a single serum sample for many different agents, and some of our biggest panels of agents would have 20 different viruses on them, we looked at multiplexing. Now, multiplexing is where you run multiple assays in a single well or subarray. So multiple assays within basically a single reaction vessel. And this can be efficient because we wouldn't need to use as much reagents. We don't need as much sample. And if we can do more controls, then we can have better accuracy. So multiplexing actually works very well for serology. It does not work very well for PCR, and I'll talk about that later. But we did pursue multiplexing for serology. We use what's called and I think some of our competitor labs do too, and it's widespread in human labs, this Luminex multi-analyte profile, or XMAP. And in this, instead of putting the antigen onto the bottom of the well in a plate, we put the antigen on these polystyrene beads. And then we can expose those beads to the animal's serum and see if the serum will bind to the antigen that's bound to the beads. And then we use a secondary antibody to show. The beauty of it, though, is that we can use different color beads. And so these beads come in sets of up to 100 different colors. So you can run 100 different assays in a single cocktail type mixture. And so in a 96-well plate, you can run 96 serum samples each for 100 different assays in theory. And so it really uses much less serum and is much more efficient for a high throughput system. Then what happens is those beads go through what's like an old fluorescent activated cell sorter, where each bead gets interrogated by two lasers. One laser checks for the color of the bead. Is it an MHV bead? Is it an MPV bead? Is it a mycoplasma bead, etc.? And the other laser checks for the phycoerythrin reporter dye to see whether or not the animal's serum bound to that particular antigen. So that one of them says yes, this is a mycoplasma bead. The other laser says, but was there anything in the serum that bound to this bead? And so for each assay, it'll check 25 to 100 beads, and then the result is the mean fluorescence index. And so it gives us an idea of the strength of reaction. And that helps also, because a weak positive is more likely to be a false positive. A very strong positive is more likely to be a true positive. So that works very well. We call that the multiplex fluorometric immunoassay, or just MFIA. So high throughput with much less volume. We use many fewer plates, whereas before we used a complex robotic array to move plates around from a washer to whatever. Now we just use this simple setup that you see in the illustration here. And by having the possibility to have all those different beads, we can use multiple antigens for some diseases. For example, MHV, I think we use 4 different antigens to screen for that, to be sure we don't miss any of the variants of MHV. And it also allows us to use more controls. So if you send us something and say it's mouse serum, we check to see whether there is mouse immunoglobulin present. And if there's no mouse immunoglobulin present, we know either you sent us something from another species, or maybe it was an immunodeficient mouse and didn't have any serum. And so instead of just giving you a negative result, we say this was not a suitable assay. And we use tissue controls and all like that. So it's really a nice system. Also, by having it much more simple, fewer things go wrong. It uses really small volumes of serum. Really, all we need to run this assay is 50 microliters of dilute serum. It's diluted 5-fold, so all that would really require would be about 20 microliters of whole serum. So it's minute amounts, and that's good. And that also permits the next advance. So because we require so little serum, do we really need you to send us fluids, whole serum, which is harder to ship? And the answer, of course, is no. And that's where we get into the dried blood spot testing. So if you just put a spot of blood onto this filter paper, we can elute out enough serum to run the assays without losing sensitivity. Because the blood gets to just dry on this filter paper, then you can just ship it at room temperature. So you don't need to worry about shipping serum with all the wet packs and styrofoam and everything else. This is an illustration of it I got from the CDC. It's an old technology. It's used a lot on human infants for some of the tests where they just take a little dot of blood from the heel of a newborn and test that. Basically, you just put the blood onto a card, take a hole punch that out, put it into a plate, soak the serum out of that, and then run that like you would normal serum. Charles River's version of it is called the Easy Spot. And so it's good because it's such nice, long-established technology. You don't have to kill animals to get enough serum to bleed. And with what we can elute out of there, you can run a wide variety of analytes. You can run some clinical chemistries as well as the antibody serology. So it allows survival blood collection. You can keep your sentinels for more than one sampling period easily. You can even test some of the animals in the colony directly without having to use sentinels. And this can be good to try to corroborate any positives that you got in the sentinels, or if you're trying to test specific animals. And then also it's a much more convenient, lower labor way to prepare and ship the samples to the diagnostic laboratory. So it seems to be catching on really widely, and I think it's a great thing. But you're still only testing one animal at a time. We found that when you dilute it out, we can get similar titers out of the dried blood spot eluates that we do out of the whole serum. So we get just as strong a signal from the dried blood spot as we do from the whole serum, which is a beautiful thing. There are some, you need to pay attention to the storage of the paper and the humidity. Obviously, the way it absorbs the blood will vary a little bit with the humidity. You don't want to put too much or too little blood onto it. If there are extreme fluctuations in hematocrit, that could allow, you have to let it dry. And then when we take the punches, and we take a couple punches from each area, from each circle, we like to select from some place where it looks like it's uniformly distributed and the blood isn't sort of piled up into one area. So there's a little more potential for variability, but we are not finding a problem. So it's a great thing. It's been around for more than 40 years, but really just now is going into rodent diagnostics. Much more convenient for shipping, and but still has the sensitivity. However, still it's just one animal at a time, and it doesn't address the basic surveillance problems with individually ventilated cages, which is that are things getting to the sentinels in the first place, or how to sample enough animals. And that's where we come to PCR. PCR panels have become a primary tool for screening groups of animals directly without the need for sentinels. So you can just take samples directly from an animal and see what they have. Using sentinels really just sees if the animal is shedding infectious amounts of material, but then you have to wait for the sentinel to get infected and maybe produce antibodies. So it's much quicker if you can take them directly from incoming animals. And that's a great thing to use during quarantine. It's faster, and it's also more sensitive because PCR can detect sub-infectious levels of shedding. I personally don't think intermittent shedding is a problem, but if animals do intermittently shed, you would think that they would sometimes shed sub-infectious amounts. It wouldn't be just all infectious or none. We do find animals that are shedding sub-infectious amounts at times. So we've been using PCR for a long time. PCR is an old story. We've been using it to screen for Helicobacter, and then we've started using it as a supplement for definitive identification of some bacteria. We use it primarily for Pasteurella pneumotropica, Streptobacillus, Corynebacterium bovis. Normally, Mycoplasma pulmonis cultures would need to go for 2 weeks. We can do PCR faster than that and with more sensitivity, so we use PCR instead of the 2-week culture for that. We use it for Campylobacter and other things that would be difficult to culture. Clostridium piliformis for Tisser's disease would be another example. So we've been developing more and more assays. And we've also used it as a confirmatory test for viral infections, primarily using mesenteric lymph node, or MLN, as a sample. And that's because mesenteric lymph node saves little bits of DNA from things that get sampled in the intestine. So mesenteric lymph nodes kind of acts like a little archive, saving bits of DNA and antigen that goes through the intestinal tract. So as we've developed more and more PCR assays and getting lots of samples through the lab, which is a good thing, the large number of samples and assays has really encouraged miniaturization and automation because we've needed to increase throughput. So how can we increase throughput? Well, one way would be if we multiplexed PCR. In other words, ran multiple PCR reactions in the same vessel at the same time. I mean, certainly that would save reagent, materials, and labor costs, and I think some diagnostic labs do it. But the problem is that if you have one agent present in high copy number, it can mask the presence of others. One can kind of hide the others. And very commonly, Helicobacter will be present in high copy number, and we know that's very common. Parvovirus is also quite common, but can be present in low copy number, and that's what's illustrated on this slide. You can see that the top row of bands on this gel is MPV that was added at 100 copies to this assay. The bottom row of bands is Helicobacter, and you can see that as the Helicobacter concentration increases going towards the left from 10 to the 0, which is 1 to 10 to 100, up to 100,000 on the far left, you can no longer see the parvovirus. MPV seems to disappear, even though it's still present in the same concentration. So the high concentration of Helicobacter masks the MPV at high concentration. And we do frequently, as you will see in later slides, find Helicobacter in the 10 to the 4 to 10 to the 5 range. So it makes it hard to find Helicobacter or to find the MPV. We do not think multiplexing is a good idea. So what do? We miniaturize the assays and doing them on an open array platform to avoid multiplexing. And this is what we use. On the left, you can see that there is just sort of draw it here, is a size of a microscope slide with many wells. There are 48 of these subarrays. Each subarray has 64 holes in it. Each hole is actually only 33 nanoliters total reaction volume. But that's aspirated into it. It's hydrophobic outside and hydrophilic inside to hold the 33 nanoliters within it. But this means that each well is one assay. So we don't have to multiplex. Whatever's present in one does not interfere with what's present in the next. So that avoids the problems of multiplexing. And it also gives us lots of wells to run a lot of assays and a lot of controls. So when it's loaded onto the slide, it looks like this. You can see that the top one has the samples loaded in, and on the bottom one you can see the fluorescence that shows when we get positive reactions. Here on the right, you can see these are the tracings that we get for each of the reactions. So cycle number goes across on the bottom. As cycle number increases, we get more and more fluorescence. And when it hits the threshold, we call that the CT value, or the cycle, the threshold for, anyway, the number of replications it took before the fluorescence hit threshold. And that allows us to estimate how many copies of the target agent are present. And that gives us more than just a plus minus. It allows us to understand what is really going on. So our panels, we developed some fairly extensive panels. These are some of the viruses that are included. We have panels that are just mouse, just rat. We have panels that are just even more viruses than this. Many different parasites, pneumocystis, a variety of protozoa, pinworms, termites. Controls go on to each one. We use a nucleic acid recovery control because we want to be sure that we're able to get enough nucleic acid out of the sample to make it a valid test. And then we also think it's really important to use the PCR spike inhibition assay to be sure that there isn't anything present in the sample which is inhibiting the PCR reaction. And that happens sometimes when you're testing feces, when you're testing material that may have alginate present in it. Sometimes a really large amount of host DNA even can inhibit that somewhat. So those are very important controls to include with each assay. They're all run in duplicate, and we also run a lot of bacteria. And then we do have other agents. So we have panels that would include ectromelia, sendai, Rio, pneumonia virus, etc. So remember this slide, looking at the limitations of soil bedding sentinels? We still haven't shown how we can go back and fix this. We've talked about that there are problems. Soil bedding sentinels don't work great. We seem to have some pretty sophisticated tools to work with. But how can we use these innovations in PCR serology to actually improve the programs? Okay, keep in mind what each test tells you. Serology, you're saying, does the animal have antibodies? In other words, was it infected sometime before last week? Serology is retrospective. PCR is just asking, is a certain small piece of DNA or RNA present? It doesn't necessarily mean it's infectious. It just means, is it present? Those tend to disappear from an animal when they produce antibodies. So the antibody response or immune response will usually clear the virus. So it will disappear from the feces. But DNA and RNA can stay in the lymph nodes and can persist in the contaminated environment. So if it gets shed in the dander or fecal dust or whatever, it will stay in the environment. So PCR can either be current by checking the feces or retrospective by looking at mesenteric lymph nodes or dust. And really I should point out, mesenteric lymph nodes will become positive usually within a couple days after infection. Dust can become positive within a week or so, and both of those will stay positive for a very long period of time. So what do we do with this? How can we actually get a better picture for what is happening in the rat currently? Well, you could do the routine sentinel serology plus some PCR. Because PCR is so sensitive, you can pool 10 fecal pellets into one sample. So you could take from the sentinel and from animals on the rack, maybe representing different breeding lines or different experiments. You could even pool between racks to manage the cost. So that'll give you a little better coverage, but still doesn't really give a 100% coverage. And that's where we get to the exhaust plenums or the filters. These are concentration points for dust and dander in the air system, the air exhaust system within the racks. So we think of that as a type of environmental monitoring, and that's not a new idea either. People have been looking at exhaust ducts or surfaces for pathogens. We've done it on barrier rooms for quite a while now. We've done it after decontamination to verify a cleaning process. People have swept up dust and used that to monitor. Sue Compton and others at Yale a number of years ago actually piped the exhaust on IVC racking through a sentinel cage and monitored that and found that it could contribute. And then more recently, people have actually started doing more targeted PCR on the plenum dust. Eric Jensen reported last January 2013 that even a single cage on a rack with a termite infestation could be detected within weeks on an otherwise completely clean rack just by testing the dust in the horizontal plenums. So it can be a sensitive and relatively rapid way to detect what's on a rack. And these are some pictures from Dr. Jensen. I forget exactly which rack manufacturer these were, and I don't want to guess wrong, but so we'll just leave that. But you can see that he is sampling in the exhaust plenum. Just we like using these sticky swabs. We like using sticky swabs. We will send those out to anybody who wants them. We find that using the optimal swabs can give a 10-fold difference in signal strength. So it makes a difference what swabs you use. And then obviously you need to use a swab that your diagnostic lab can then retrieve the nucleic acid from. So current evidence suggests, as Eric and others have found, that it's very sensitive for mites and pinworms. Very good strong signals for Helicobacter, Pasteurella, Spironucleus. You can get good signals for parvovirus, norovirus, rotavirus, lots of different bacteria, Corynebacterium bulbus, Cryptosporidium, pneumocystis. Really, it seems to be working very well for a really wide range of pathogens. Here is one example. This rack had been in use for about a year. We started looking because somebody wanted us to look for parvovirus actually in the plenum dust. And so we checked the plenum of dust for different rows. And you can see that we did find parvovirus. But since we were running it on a panel that had other agents, we went ahead and ran it for a lot of different agents. I would like to take a look here at the number of copies that we were finding for Helicobacter. This is 100,000 copies, 10 to the 5. This is the level where 100 copies of parvovirus could be inhibited, might disappear if we were doing multiplexing. So if you're doing multiplexing, you might not have found these if you also were checking for Helicobacter at the same time. And these lines, the red lines, indicate agents, I think, that were a surprise to the people doing the racks, things that were not being picked up by their soil bedding sentinels. It's very rare that Pasteurella pneumotropica gets picked up by soil bedding sentinels, even though they're readily detected by PCR on the plenums. This is a real extreme example. Instead of taking it from the exhaust plenum from an individual rack, we went to the cage wash area, the bedding dump station for a centralized cage wash for the entire facility. Now, I think they had dogs and monkeys and different things in the facility, so it wasn't just mice. But just by sampling the dust at the dump station, we all look at everything that we found here. The ones again, underlined in red are things that are not likely to be picked up by the soil bedding sentinels. But you can see also, probably from some of those other species, we were even able to pick up things like salmonella, etc. So really, you can find just about anything that's present in the soil bedding. So remember this slide? You can probably guess where I'm going with this. Soil bedding sentinels are pretty poor at picking up a lot of the common infections, but we find that they are very readily detected by rack swabs of the plenum dust on the types of racks where the dust will accumulate in those plenums. So where are we now? Keep in mind that not all racks are the same. They're not all equal. They may all work all equally well, as far as I know, for containing infection now, keeping it from spreading cage to cage, but not as far as the plenum sampling goes. Some of them have filtering on the exhaust at the cage level, and in those cases you don't find much dust in the exhaust system. So that really makes a difference. You have to look at your individual rack and see where the dust is accumulating. If it's the filter or the pre-filter, take the samples there. You have to look where the dust is accumulating, or as we say, dust is a must. If you do have an infection and you're trying to clean up from that, then perhaps residual nucleic acid could be an issue. It's probably good to do some baseline testing in those cases, but that's only after contamination, and we're not really finding that to be much of a problem. Also, for all agents at this point, what's the minimum? If something was in only one cage at a low level, how sure are we that it would be picked up? Well, everything we see seems to be that it still works better than the bedding sentinels. So whether or not it's perfect, it seems to be better than bedding sentinels. Conclusions at this time, it really seems to be the best sample there is and completely sufficient for testing racks for mites and pinworms if the dust is accumulating. Definitely better for sentinels and better than sentinels for Helicobacter, Pasteurella pneumotropica, probably or possibly better than norovirus. Also, when we get looking at those, we find a fair amount of mouse adenovirus, cryptosporidium, things that really were surprises that we didn't realize were common. At this point, it's we're really finding a lot of that it's very good. It's as good or better than bedding sentinels for all agents that we've come across so far. When we've actually put animals from a pet shop into a rack just to see what we could find, we've even found positives for sendi, which we don't think would ever get to into a soil bedding sentinel. So to date, we have found no agent, nothing, that's detected better by soil bedding sentinels than by the exhaust air dust. Exhaust air dust seems to be as good at or better than bedding sentinels for everything so far. Somebody asked if you have everything in open top caging, how good would the exhaust for the entire room be? And we've looked at that with our barrier rooms and found that it is still a sensitive way of monitoring those barrier rooms. But our barrier rooms might have 10,000 cages and more than 100,000 animals with a lot of breeding going on. So if you only have a small number of cages, it's hard to really say if that's sufficiently specific. However, And even in those cases, if you're submitting anything for PCR, I would include a swab of the dust of those exhaust vents as a supplemental sample. If you send it to us, we can pool a lot of different samples into one assay, so one price, right? One assay. You could send us 10 swabs, 10 fecal pellets. We would extract the nucleic acid from those separately. We'd like you to send them in separate containers, but then we would pool that together for the assay. And so if I was doing it, I would include the swabs of the exhaust grades. So, additional considerations. Some things just aren't shed in the feces, such as lactate dehydrogenase elevating virus, mouse cytomegalovirus. You know, those things we really wouldn't expect to show up. But they're not going to show up in soil bedding sentinels either. And so, you know, if you're concerned about those, maybe you could do some survival bleeds with the easy spot and, you know, might be able to pick it up that way. Also, yeah, it may not be applicable to all rack systems. You'd have to take a look at, you know, your own racks, see where the dust is accumulating. You know, different manufacturers are taking a look at this now. They seem convinced that it's a good way of sampling. They sort of sense the shift in the field and are looking at adapting rack design to exhaust air dust sampling. And really, the confidence in the results depends on, you have to have confidence in your lab. You know, you need to be sure that they're using quantitative methods, have confidence in the sensitivity of their assays, controls, and all. So you have to have confidence in your lab in order to go forward with it. Sometimes we can find trace DNA without infections. It doesn't happen often. It happens once in a while. Could be cross-contamination of samples. I mean, on the sampling end, you'd have to be sure to keep, to not get any cross-contamination between samples. We go to great lengths to try to separate on our end, but, you know, it's always theoretically possible, could happen. We have seen instances where it seems there is non-infectious DNA present in feed or other bulk materials. You know, there could be DNA present without it being evidence of infection. And, you know, if you had a past contamination, you would wonder, could it be residual DNA from past contaminations that way? So you would need to go back that way. Always call your lab regarding any unexpected or anomalous results, right? If you don't think you have Pasteurella pneumotropica, you get a positive for it, or for parvovirus or whatever, before you take action, call your lab and discuss it. One of the beauties of TACMAN PCR is that it is semi-quantitative. So if you talk to us, we can often help work out whether it's likely to be consistent with an active infection or not. Helicobacter, I keep saying, is present in high copy number. If we got just a trace amount of it on a sample that you sent us, it would be more likely to be a cross-contamination somehow than an active infection. So we'll work with you on that. In some diagnostic labs, I know we will, and I would hope others would, will perform some additional testing at no charge to follow up on the initial positive. So we wouldn't leave you hanging. We got to help you work out in those cases. So if you get an anomalous or an unexpected result, call the lab. One thing I don't have here, but I always try to remind people is if you're using a diagnostic lab, you're already paying for their experts, right? You're already paying their salaries. So if you're using them, give them a call and ask questions. You know, don't ever hesitate to ask them. They also know what's happening. I mean, maybe there's a nationwide outbreak of rotavirus. If so, that would be some additional perspective on your results that it might be useful to know. So always call your lab. And so taking all of this, can the shipping of live animals for routine surveillance be replaced by just shipping serum or PCR samples? And obviously, I think the answer is yes, or I wouldn't be presenting this, but it's yes if the mice are being tested to see if they meet specifications, you know, a defined exclusion list. So if you're testing them to see if they have any of a list of 20 agents, we or other diagnostic labs could develop tests, or probably have them already, to test for those agents. And for almost all of them as far as I know, testing, you don't need to submit live animals. But it's not a vague phenotyping kind of thing. It's, do the animals have one of these list of agents? So shipping live animals is expensive. I'm sure the listeners here know that much better than I do. Shipping one crate can be hundreds of dollars, more than the cost of the testing. There are weather embargoes. I mean, much of the country right now is colder than 20 Fahrenheit, and much of the rest of the year, a lot of it is warmer than 85. So there are weather embargoes to shipping, and the microbial status can even change during shipping. So have to keep that in mind. Animals can die if they're sick. But don't hesitate to send dead animals, or don't worry too much. It's a welfare issue if they die during shipping, but you can still sometimes get some valuable pathology results from it. So, you know, if an abscess or a tumor is there, you can still see it even if the animal's pretty rotten. So don't throw away animals just because they're dead. But there is animal welfare issues with shipping, and so it's better not to ship. Ship only when you have to. The default shipment should be serum, feces, or other specimens. Live animals, you know, when would they have advantages? Well, some people find it easier just to put animals into a crate, ship them to the lab, than to take all the samples themselves. But I think if you're collecting serum, easy spot is probably easier than even putting them into the crate. Dust swabs are easy. Shipping them uses the expertise of the diagnostic lab more fully. So if it's some sort of tricky diagnostic situation, it may make sense to send live animals so that the diagnostic experts can really take a look at it. And sometimes if you're trying to culture for anaerobes, it's easier to do it by culture. We do have, we're developing PCR. There is PCR for trichomonads now, but if you're just sending feces for parasitology, remember that some protozoa don't form cysts. So we've pretty well covered this. So what would you ship? If you're shipping live mice, obviously ship the live mice. But if you want to ship samples instead of animals, you could ship serum or the easy spot. And Charles River or other diagnostic labs, I'm sure, but you know, we'll send you the shipping materials. We'll send you instructions on how to fill them out, how to complete it, what to do, walk you through it. We've got online support for that. Presumably other diagnostic labs would also. So if you're shipping samples, you could just ship serum and a fecal pellet, and really that covers just about everything. It probably wouldn't cover fur mites, so if you were looking for fur mites, probably would want to include a fur swab, or sometimes we call it a body swab. Remember that samples for PCR can be pooled from up to 10 animals. So if you're going to send a pellet from one animal, you might as well collect a pellet from 9 others and send it. It doesn't hurt the sensitivity, and it gives you a little broader screening. So if you can pool 10 to 1, I would say you should always pool 10 to 1. And remember that along in another envelope, but as part of the same diagnostic test panel, we could include 10 swabs. So you might as well send 10 swabs at the same time. Or instead of sending those, you could send pooled exhaust air duct swabs and maybe some pooled fecal pellets. Or really, pretty much you could find about anything you wanted with just those exhaust air duct swabs without the serum or fur swabs. For anything you'd use fur swabs for, the plenum swabs or the exhaust air dust swabs are better. So fur swabs really are not as necessary as they used to be. The dust swabs are a beautiful replacement for those. So just to conclude, and then we'll get on to some of the questions, and hopefully that'll stimulate even more discussion. You know, we're continuing to discover additional subclinical infections. As our tests improve, we find more and more. I don't think any of them are really new. I think they've all been around forever. We're just finding them, but it's important to know what's there. Advances in diagnostic methods now make it possible to test whether or not your in-house animals are within specifications without the need to routinely ship any live animals for health surveillance. Remember, diagnostics investigations, you know, if you have sick animals, if you have an outbreak, anomalous research results, diagnostic investigations may still benefit from submission of live animals to your diagnostic lab. And the easy spot serology and exhaust air dust PCR can reduce or replace much of the sentinel usage. So thank you. I'll get on to the questions. Just like to point out, this is where I live, right about here in the Willamette Valley. Actually, it's about over here. Beautiful wine country, and this is Mount Hood, as we can see from near here. So thank you for your attention. The questions are, let's see, I believe we talked about the room's HEPA exhaust. The answer is yes, although we don't really know the full sensitivity on it. Information on the type of swab needed. We will send those free of charge. Just contact Charles River. It's crl.com. You can just look up our website and go to the diagnostic testing website, and people will help you on that. How long does the dust sample remain viable, and how far back can the testing reach? That's a good question. The dust sample, of course, isn't truly viable. What we're looking for is genetic or DNA lengths that are long enough, sequences that are long enough for our PCR. And TaqMan uses really short sequences. They should stay present. It would be years probably before they would degrade. Potentially, they could sort of wash out with the continued airflow over a period of months. But within quarterly monitoring, we have not seen that be a problem. We haven't really tracked out low-level contaminations within a rack and then followed them for a year or so to see how long they would go. But certainly within quarterly, probably even within 6-month monitoring, we haven't seen a problem. Next question is, we may get some wild-caught kangaroo rats. Cool. I think that would be pretty interesting. And we need to check those for murine viruses. Can we use normal rat panels, or do we need to have the rat species, sentinels, and test those? I think those are two separate questions. One would be, what infections do the kangaroo rats have that could infect them or affect them? And the second question is, do they have anything that could infect rats elsewhere in the facility, the true Rattus norvegicus? And so I think it'd be better to have the offline discussion on that. Sometimes it would be useful to use rat sentinels, which could give a good answer as to whether the kangaroo rats have anything which theoretically could transfer to rats. And then we would typically use some targeted PCR for zoonotic agents to screen the kangaroo rats. We also could do serology using a G protein instead of a secondary antibody to look for sort of entire groups of viruses like generic parvoviruses, generic coronaviruses, rotaviruses, etc. But you can see that gets a little technical. So crl.com or ask CRL, and we could get into more detail on that. Quality, let's see, wouldn't it be much cheaper to send the animals for the health monitoring plus than to do all the individual tests, as of the serum, fecal, etc.? That's a good question, because I wouldn't even advise doing this if it raised your health monitoring costs. But here's how that goes. Health monitoring on one animal costs about 1.5 times a full serology panel, and full PCR panel costs about 1.5 times what the a live animal submission would be. So a full PCR panel would be about 1.5 times as much as live animal submission. But you could check 10 animals. And what would you submit for the single animal? Would it be a sentinel? If you submit a sentinel, we know that there are real disadvantages to that. You'd be better off running you would be better off running a PCR panel on swabs from 2 racks pooled. It would be less than doing 2 live animal sentinels, and you would get more information. So you got to look at your own, your own systems. It may not be right for all situations, but it's not necessarily more expensive. I mean, I think that's a good question. You have to look at it. Frequency of survey testing for most often used in rats or mice. I think most people are looking quarterly. Some places look every 6 months. There's really nothing magic about it. If Some people use 3 and some people use 6. I've heard of others using 4 months. There's really no magic number. I'd say the most common is quarterly. Falaza in Europe recommends quarterly, but there is no, we check our own colonies every 2 weeks one way or another, but it's a different situation. There is no magic answer. Soil bedding sentinels get a little less susceptible to infection after 3 months or so. So you may be a little less likely to pick up rotavirus or some others, although there's controversy on that. Some people think even that doesn't matter. So just no magic answer, sorry. Let's see. Some labs may still need import-export health certification to ship samples across international borders. That's true. I think in those cases, what we usually, we'll read the rest of it. In Northern Ireland, I had to set up rodent samples only lab referral because the available airlines wouldn't ship live animals anyway. Yeah, that's a factor. Shipping live animals can be a real problem in many cases. It's often much easier to ship samples. We can arrange to receive PCR samples that have been inactivated by one means or another, or serum samples that have been. So it may be easier to ship those. Sometimes the question comes up about, for international shipping, certain long lists of agents may be required. But I think usually you're better off only testing the animals destined or considered for shipping for that really long list, rather than your entire facility all the time. But of course, you know, you have to resolve that for your own facility. Question is that the cost of health certification shipping of live animals can be prohibited. Absolutely. It's illegal to ship animals likely to give birth and sucklings in the UK. I think that's a fact probably, but it also makes sense to me. So if you want to monitor samples from those, then it's best to ship the samples. And so that may be the only method available in some cases. But I think for all, if you're looking against a set list of agents, the testing is at least as sensitive as shipping live animals anyway. So you're really not giving up any, any testing sensitivity. Question, ask your state veterinary service about the licensing, particularly if you want to send live placental mammals to Australia. And yes, I would always recommend before shipping any animals of any kind anywhere to be absolutely sure that you're doing it within the regulatory guidance. You know, that's the last thing anybody would want to be caught with. And in some situations, the same thing for samples from living animals. So, samples collected from animals, sometimes CITES permits or whatever are required. So everybody needs to be careful about that. Question is, can we do an assessment plus with the easy spot? And the answer is yes. So we can do any of the assays with the easy spot, which is our name for the dried blood spot systems. So any other questions at this time? There was one question which I dodged, admittedly, on which QA or ISO program do we subscribe to, and I don't have an answer for that. I don't know the answer for it, but if you contact me offline, it's charles.clifford at crl.com, charles.clifford at crl.com. We can find you an answer. So thank you very much, everybody. If there are no more questions, we can close. Thank you very much.